This part is going to be very, I think, okay, for biochemistry, very, very important because it actually gives you a lot of the information that you need to understand enzyme and enzyme kinetics. Okay, so here we're going to talk about hemoglobin and allostery. Okay, and actually I'm not so interested in hemoglobin. I'm actually more interested in allostery. Okay, and I'll tell you what that is. So let's come back to our picture. Right? This guy is myoglobin. It's a monomer. Hemoglobin is a heterotetramer. Okay, so it's going to be doing a lot more than what myoglobin can do. Okay, so it can do a lot. So let's coming back to our O2 binding curve. So again, the top line is myoglobin, and we say hemoglobin or any other transport protein have to have a lower binding affinity than your myoglobin. Okay, and then we saw in the previous lecture that you cannot have a efficient fixed affinity transport protein. You also don't want to have a fixed affinity but low affinity protein for as your transport protein. So either too high or too low. It's no good if the affinity is fixed. Okay, so what we want to do is we want to have a protein that can change O2 affinity depending on the location. Okay, so here we want to have weak binding at low O2 concentration. Okay, we want to have weak binding at low because if we have weak binding at low, then that will give us a lot of advantage to unload oxygen onto myoglobin, right? Because if this guy is weak binding and myoglobin is like super strong binding, then I have a large gap, and then this gap can be used more efficiently. Okay, so you can deliver more packet. Now, when the O2 concentration is high, okay, so around this region, you want to have a strong binding because you want to have a very powerful delivery person that can carry a lot of stuff. Okay, so you want to have strong binding at high PO2 and low weak binding at low PO2. Okay, so that would be an ideal protein. Okay, and this is sort of what we call cooperative binding curve. Okay, because the subunits or the proteins can have cooperativity. Cooperativity meaning that you can sort of change behavior dependent on the stuff that you're loading onto it. Efficiency in O2 transport is achieved by cooperative binding in multi-site protein. And this is described by a sigmoidal binding curve. Okay, so because like that. So initially the slope is very small, and then all of a sudden it transitions. You have a transition from weak to strong binding site. So initially it's weak, and then you have a rapid transition, and then it becomes strong. Now, it is important for hemoglobin to bind O2 efficiently at high O2 concentration places such as the lung, but low binding efficiency at low PO2 places such as muscles and tissues. Now, here's a question. How can it achieve this? How to selectively change binding efficiency? Okay, protein structure, if it's fixed, how do you change this ability? So this is where I introduce you guys to the idea of allosteric effect. The cooperative binding of oxygen by hemoglobin is one example of what is referred to as allosteric effect. Have you guys heard about allosteric effect previously? This is actually a very important concept because after you go to higher level biochemistry courses or higher level molecular bio classes or whatever, there's a lot of proteins, okay, proteins in your body are a lot of times regulated by allosteric effect. In allosteric binding, the uptake of one ligand by a protein influences the affinities of the remaining unfilled binding site. So this is sort of the definition, or this is the description for hemoglobin. So if your molecular oxygen is bound on a hemoglobin, then it's going to increase the affinity of hemoglobin to bind more oxygen. Now, a couple of things to help you remember. Allosteric, allo means other, okay? Cereals meaning solid. So allosteric means affecting the activity or function of a protein or enzyme by the binding of a molecule on a position different from its active site. Different from its active site. So this is sort of the key, right? This is actually very important. Allosteric binding means that you have a protein. Proteins have active sites. Okay, we know that. Proteins have active sites. Those are the location in which catalysis occur or O2 binding occurs. That's the active site. Okay, but allosteric means that it's a different site, a different location in which if a molecule or a ligand bind to that protein, it will cause the protein to undergo some kind of a conformational change, and then it's going to alter the protein's function, okay? Either by inhibiting it or activating it. Now, here it says the ligands may be of the same kind, as in the O2 binding to hemoglobin, or they may be different. Okay, so allosteric, the ligand for allosteric effect does not have to be different molecules. It could be the same molecule, it could be different molecule, whatever. As long as you guys understand the idea of allosteric effect, okay, it's some location outside of the active site of the subunit, I forgot to mention, of the subunit. In the case of hemoglobin, what you'll see is that O2 binding is at the active site of one subunit, but it changes something, it causes the other subunit to increase in affinity. We'll get there, okay? All right, but, so hear me out. So in this case, the ligand in which causes your allosteric effect could be the same as your substrate, it could be different compounds, it could be whatever. So allostery is an important mechanism for regulating the activities of enzyme. This is actually very important, right? So if you think about it, what are some mechanisms that we can use to regulate cellular activities? Regulation. 比如说你今天 maybe your blood glucose is low, okay, and after you eat something, then it's going to cause a metabolic state change. What are some mechanisms in which you can achieve metabolic control or cellular control? You have transcription, right? Go from DNA to RNA. You can inhibit transcription, or you can activate the expression of a gene. Sure. Okay. What else? What else can you do? Maybe inhibit the translation. So if you want to control something or regulate something, you can do it via transcriptional control, translational control. However, what's the problem in those cases? Maybe too slow, right? Because what if I want a change immediately and then the enzyme is already there? Okay, how do I turn off an enzyme by stopping the translation or transcription? I can't. The enzyme is already there. So this is where allosteric effect is very important, because what allosteric effect can do is it immediately shut off your enzyme. Okay? It doesn't have to do it. You don't have to achieve control through transcription or translation. You can directly affect the enzyme that's already there. So you can have a rapid response. Okay, so that's what allostery is very important for. All right, so let's come back and talk a little bit about cooperativity. Cooperativity is referring to a conformational switch from a weak binding state to a strong binding state. And in this case, this is talking about hemoglobin. Okay, and essentially you have a protein tertiary structural change. And in this case, I wouldn't exactly say tertiary, so it's essentially a protein structural change. Okay, so you have a change in protein structure that caused affinity to be different. All right, now cooperativity in binding requires communication between the binding sites, okay, or between different locations of the overall protein. Hemoglobin has 4 subunits. Okay, that means that you have to have some kind of a communication between the 4 subunits. Okay? So here we have the Hill equation. Don't worry too much about where I got this Hill equation or anything like that. All right? Just as long as you can tell me, as long as you know that you have here H is equal to Hill coefficient, okay? And then you plot this log of bound over the unbound, okay? And you plot that versus log of PO2. Well, essentially, that's just like you take your concentration, okay? And then versus your total amount of the fraction of the O2 bound, or whatever. Now, essentially, all lines at a stable state, okay, at a state, whether low affinity, high affinity, medium affinity, whatever, at any state, you will have a linear slope, okay? You will have a linear slope. Here, it says that myoglobin has no cooperativity and therefore is a linear line with slope of 1, okay? It's a slope of 1. But in the case of hemoglobin, what you can see is how it's not a linear line, right? It's not a linear line. It changed the x, not the x-axis. You changed you had a translation upwards, okay? So you have a translation upwards. But what you can see is that towards the end, the slope is still equals to 1. Okay? Now, what the Hill constant, okay, or the Hill coefficient, what Hill coefficient is what is the slope, okay? What is the slope when this thing is equal to 0, okay? When the slope, okay, the slope of your curve at that location, okay? Now, myoglobin, the entire line is y equals to x, okay? Slope is 1, okay? But for the red line here, the slope is not 1, right? Because it's essentially that, okay? So this is y equals to maybe 3x, something like that, okay? And then Hill coefficient is talking about this 3. So for both cooperative and non-cooperative systems, the Hill plot gives the value of H, Hill coefficient, as the slope at this particular location, okay? Now, H, you can have 4 different scenarios. If H is equal to 1, that means that there is no interaction between the sites. Why? Because what you can see is that the stable states, okay, the stable state, the high affinity state, slope is 1, low affinity state, slope is 1. So if it's stable, okay, meaning that your protein will not change affinity or it does not change anything, slope is equal to 1, okay? So if H is equal to 1, it means that here nothing is changing, okay? So it means that there's no interaction between the sites. Therefore, the molecule binds ligand non-cooperatively, okay? For example, myoglobin, okay? Now, another situation, okay? So hemoglobin, this is hemoglobin, okay? Is that your H is bigger than 1, lower than N, okay? And I'll tell you what that N is just a minute, okay? It means that there is interaction between the sites is the number of binding sites, okay? All right, so if your Hill coefficient is larger than 1, that means that this situation is the usual one for a protein that binds ligand with so-called positive cooperativity. Okay, so if H is larger than 1, then you have a positive cooperativity, meaning that the binding of ligands will increase the affinity, okay, the affinity for other substrates of this protein. Okay, so the binding of a substrate increases the affinity of this protein. Okay, now here, this one, number 3, H is equal to N. This is the energy of interaction between sites approaches infinity in this hypothetical. This is hypothetical. There's no enzyme that has H equals to N, okay? In this hypothetical situation, the molecule is wholly or infinitely cooperative. Okay, infinitely cooperative. Right? This is the upper limit for cooperativity. You cannot have H larger than N. That's not possible because if you only have 4 subunits, okay, or 4 binding sites, you cannot have cooperativity larger than 4. Right? It just doesn't make sense. You don't have a 5th secret binding site. No, it doesn't exist. Okay, there's no such thing. Right? So you cannot have H larger than N. But when H is equal to N, that's also a hypothetical situation. No enzyme will have a 100% completely and infinitely cooperative behavior. Doesn't exist. Okay? Now, H less than 1, that happens sometimes. In this case, ligand binding at one site reduces the binding affinity at other binding sites. This is called the negative cooperativity. Essentially, it means that after you bind one substrate, the protein's binding affinity decreases. Okay, so really, you have this one is no cooperativity. This one is positive cooperativity. And here you have a negative cooperativity. Okay? So essentially, that's it. Okay? It's really quite simple. Right? Don't worry, this is not super important. I'm more interested in you understanding the principles. Okay? All right. So here we're going to say hemoglobin somehow can switch between conformational states with lower and higher O2 binding affinities, right? We saw that it was like that, right? So it changed affinity from low to high. So generally speaking, we'll define, so this term is actually used a lot, right? So we'll call that you have a tense state, T state, okay, when O2 is not bound, okay, and the binding affinity for O2 is weak. This is referring to the deoxyhemoglobin. Okay, so we have a couple of things. T state is equal to deoxyhemoglobin is equal to low O2 binding affinity. Okay, 3 things. T state, deoxyhemoglobin, and low affinity. Okay? Now, on the other hand, you have the R state. Okay? We call that the relaxed state. Relaxed state when O2 is bound. Okay? And the binding affinity for O2 is strong. This is the oxyhemoglobin. Okay? So we have T state, deoxyhemoglobin, low affinity. R state, oxyhemoglobin, high affinity. At low O2 concentration, hemoglobin is at the T state. Okay? And as O2 concentration increases, hemoglobin will turn into R state. Okay? So as a function of oxygen presence or concentration, your protein will shape shift. Okay? Right? Like magic. Now, essentially, in scientific terms, okay, it says that oxygenation causes hemoglobin quaternary structure to change. Okay? So nothing's magic. Essentially, what happens is you have one alpha beta dimer rotate and slice with respect of the other. You don't know what that means, so I'm going to show you a picture. Okay? Binding of O2 in the T state. T state is the deoxy, remember? Okay? T state is the deoxy, so low affinity. So binding of O2 in T state triggers a conformation change, and then you change the protein into the R state. Okay? The subunit structure changes very little. Okay? What you actually have is a conformation change by rotation. So it's just a rotation, right? Now, here, remember, I think it was last lecture or 2 lectures ago, I forgot, when we talked about quaternary structure, remember we had that point group thing, right? That C2, axis of symmetry, C3, blah, 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 that kind of stuff, right? Remember, I didn't want you to like memorize all of that. I just want you to know what it is, okay? So in this case, the axis of symmetry is from top down. This is the axis of symmetry. So this is a two-fold axis. So this guy is a C2. Minor, doesn't matter. Okay? Now, so what you actually get is after you bind oxygen to hemoglobin, what you have is a slight rotation from alpha 1, alpha 2, slight rotate, okay, by about 15 degrees relative to the, alpha 1, beta 2, relative to the alpha 2, beta 1. Okay, so you have a sort of a sliding occur. Okay? And what the sliding does for you is that it's going to cause some changes. Okay, so this picture right here, this is from essentially a side view. Okay, so this is the axis of symmetry is like that, and then we're looking at it from the side. Okay, so if we look from the top down, okay, so this is top down, this is the axis of symmetry going in and out of the picture. Okay, what you notice, is that you have a slight shrinking of the center. Here, the shift from the deoxy to the oxy state is evident, meaning that you can see it obviously by the shrinkage of the central cavity and the shift in the contact of residue with the chain. Okay? What does that mean? Okay? Look, initially you have a larger hole here, now you have a smaller hole here. Okay? Another thing that changed. Initially you have a bunch of residues here that interact. Now they are separated. Okay? So something happened. Right? Now, turns out these things, these interactions are salt bridges that are used to stabilize your T state. Okay? They're actually used to stabilize your T state. So what you see is that here in the T state, you have initially a bunch of these salt bridges here, interactions, okay? So you got a bunch of these salt bridges that sort of put the alpha and beta together, and they have very good interaction, okay? So T state is stabilized by the salt bridge. R state, when you pop it into the R state, what you see is that, whoa, they're really far apart now, okay? They're like not very happy. You lost the salt bridges in the R state. Is this a favorable or a not favorable process? It's unfavorable, right? Right? Remember we said the formation of salt bridge is very stable. So here you broke the salt bridges. That means what? You have to put some energy in the system somehow. So the question is, if T state is really stable, then why does hemoglobin change to R state when O2 binds? That's a very legit question. You should be curious about that. Turns out, T state is not exactly very, very stable. T state is stabilized by the salt bridge, yes. Okay. However, the heme in the T state, remember T state is the deoxy. Okay. Remember we went through the whole thing about your blood being red and your blood being purple, right? So actually in the T state, because you don't have that molecular oxygen on your heme is going to be tilted. Okay, so see, your heme is tilted. Does your heme like to be tilted? No. Okay, why? Heme is a what kind of molecule? Aromatic. Aromatic molecule wants to be flat. And then all of a sudden you're taking a flat molecule and then you're like bending it. Okay, you're bending it. So there's energy stored in here. Make sense? Right? Makes sense, right? Because it's like you taking benzene and then you're bending it. That doesn't make sense, okay? So it can't work that way. Here you have the deoxyhemoglobin. The heme is bent, okay? Now, when you throw oxygen into this system, what happens? What happens? Okay? Your heme pops back into the planar position, okay? Your heme is now flat and happy. Okay, and happy. Now, in this process, the heme here the right arm of the heme is going to hit a valine. Okay, the heme bumps into a valine, and the valine says, "Yo, I'm not happy." So the valine has to rotate, and valine rotates. The histidine also rotates. Why? Because they're the same axis. Okay, but essentially they're the same. They're not the same axis. They're the same helix. Okay, they're from the same molecule essentially. Right. So after the heme hits valine, valine says, "I'm unhappy." So valine moves. Valine moves. The histidine moves with it. As a result, you have a rotation. Okay, you have a rotation to the molecule. All right. So this is the summary of what I said. When O2 is absent, the heme is puckered. Okay, this is called puckered. And the binding of O2 to the oxyhemoglobin causes a conformational change to the heme such that it becomes planar. And then binding of this O2 ligand pulls the iron into the heme plane, which flattens out the heme and causing a strain between the valine and the heme. Okay? As a result, you have a shift in orientation of the histidine, this guy, okay? In addition to the valine, okay, because the valine is pushed to the right, and then your histidine also moves with it, and then together you have a structural change, and then that causes the salt bridge to break. So naturally, originally your F helix is tilted, right? It's tilted, okay? And then when O2 is added to the system, obviously you see how the heme becomes flat, and then as a result, your F helix also changes. You have an angle, right? A changed angle. Okay, so you have a movement of the F helix. Now, how do we know this? Okay, so we can use site-directed mutagenesis. Okay, meaning that we can, because we know the DNA for making hemoglobin. So what we can do is we can alter the DNA of the hemoglobin, express it in E. coli. Okay, so I can change the genetic code for hemoglobin, and then I change the proximal histidine. Okay, so FA histidine. This guy, remember, is the proximal histidine. I change the histidine into a glycine. Okay, so now that your F helix is completely not associated to the heme. Okay, so then. And you'd be like, whoa, but my heme is not happy without histidine. So what then we can do is add imidazole. This guy is imidazole. Okay, that's imidazole. That's the functional group from histidine. We add imidazole, and then imidazole will be attached to the hemoglobin. Okay. Now, lacking a connection to the heme, what you see is that then the F helix is not disturbed by O2, and then there's a significant reduce in cooperativity. Okay, so what you see is that the rotation of the F helix is due to the conformational change of the heme. Okay, so heme initially unhappy, now happy. We can have a simplified view of ligand binding and conformation energies in hemoglobin. So we have increasing in O2 concentration from 0 to 1. Okay, initially, at 0 O2, the T state is more stable. Therefore, this is delta G. Okay? T state is more stable. Why? Because you have a bunch of salt bridges, and having salt bridge is sort of links the proteins together, and then everybody is happy. Okay, even though your heme is not happy, that's fine. Okay. R state at low O2 is going to be higher energy. Why? Why O2? So the fraction of oxygen bound to the hemoglobin increases. The energy provided by the formation of the iron O2 bond stabilizes the R conformation relative to the T conformation. So O2 binds to the iron goes into the center of the heme becomes happy, and then kick out the valine, then it's reversed. Okay, so R is stable. So what do you want to know? The relative change. So T, okay, so the delta G's are quite different, okay, because you're the heme business. Okay, so the energetic cost of breaking stabilizing interactions in the deoxy state is paid by the formation of the iron-oxygen bonds in the oxy state. All right, now, corruptivity. There are two models. Okay, I'm going to tell you right now, the real process is neither of the two models. It's a combination of the two models. Okay, so these are like the two extreme models, but the real process is a combination of these two. Okay, I'm going to tell you why. All right, so we have two. One is sequential model. This one is symmetry model. A lot of time it's also called concerted model. Essentially, what happens is this. All right, so in both cases, we have hemoglobin subunit in low affinity, hemoglobin subunit in high affinity. So one is essentially giving you a square, the other one gives you a circle. Okay? Now, you have essentially the T state and R state. T state being square, R state being a circle. Okay? Now, the little dot here represents molecular oxygen. Now, sequential model is, in a way, okay, more similar to my description so far. Okay, so under the T state, so no oxygen present, little oxygen present, you are primarily in the T state. Okay, so your protein is in the T state. Okay, now as the oxygen bind to the molecule, okay, you start to form these intermediates. So these are like intermediate structures. Start to form intermediate structures, okay, and then as the oxygen eventually increase, okay, you have different intermediate structures, and then at places like this, you have, for example, here is 3 bound, 1 unbound, and then you still have intermediate structure. Okay. Now, so you looking at this model. Okay, looking at this model, in a way, kind of what you guys expect, right? Because one subunit bind, I change conformation, and then I start to bind other subunits, right? However, through experiments, people know that even without 100% binding, you can still have a hemoglobin that's in R state, complete R state without 100% binding. So things like that should not exist. 就是用实验，有些人用实验证明 ，OK， 你 actually 可以用实验证明说，当你今天有这个 hemoglobin， 它不是在100% 的 saturation，OK， 也不用到很高的 saturation，OK， 然后呢，他们就已经发现说，有一些已经是完整的 R state， 所以它不是这种没有这种太多的 intermediate state， 它没有这种太多的 intermediate state。那上面的 sequential model 基本上是告诉你，你有一堆 intermediate state。OK，but 实验告诉我们说不太像这样。OK， 基本上 T state、R state。OK， 那可能有一些中间的 intermediate state， 但是基本上就 T state、R state。Now, the concerted model or the symmetry model then says at low O2 concentration, you already have the T state and the R state. It's just that the equilibrium between T state and the R state favors the T state. Okay, you still have R state. You still do. Okay, you still do, but maybe very little. Okay, but we know that that's not quite true either, because if you don't have any oxygen on the protein, you don't really have R state. Okay, it's primarily just in the T state. Right, so that's also a little weird. But let's take a look. Okay, so here what you have is this is T state, R state, and then of course the R state one bind oxygen more favorably, so you first generate a bunch of these intermediate states that have the R state filled with oxygen, okay? And then you fill the R states with a bunch of oxygen, and then you're shifting the equilibrium between the T state to the R state. Now, the concerted with a symmetry model also is not exactly accurate, because proteins, you don't really have an equilibrium without oxygen, right? You only turn into the R state once you have oxygen bound inside of the heme, right? But here it says, nah, T state and R state just, we have an equilibrium going on. Okay, so that's not exactly true either. Okay? So the truth out there. So the real mechanism is a combination of both, because we cannot explain with only one model. Okay, so we have to explain hemoglobin using both models. So going beyond simple T and R state, turns out hemoglobin is this really, really amazing protein. It has all sorts of other stuff. Okay, all sorts of other stuff. So we know oxygen. Okay, oxygen is both the cargo and it's the activator for hemoglobin to change affinity. So oxygen is a thing. Now, cooperative binding and transport of oxygen are only part of the allosteric behavior of hemoglobin. So hemoglobin actually has other allosteric effects, okay, not just by molecular oxygen. Okay, complexity of living organism requires that proteins and enzymes be flexible to adjust to different conditions, parentheses, environments. Because you are a complicated organism, and you are going to be in different conditions at different times. Your body or your cell has to be able to adjust to these environments. So here we'll look at some of the allosteric effectors. Okay, allosteric effectors meaning molecules that will have allosteric effects on hemoglobin. Now, some of the examples include pH. Simple as that. Okay, pH. So protons. Protons are going to affect your O2 binding. Okay, you have chloride anion. You also have this little molecule called 2,3-bisphosphoglycerate, 2,3-BPG. So we got some things going on. Now, many enzymes have allosteric regulators for on-off and increasing, decreasing their activities for quick control of their reactions and functions, like what I mentioned earlier. Now, a couple of key terms that I want to cover. We have active site. Okay, so I already talked about active site. Active site is a particular site or location on a protein where the protein must bind one or more substrate molecules to carry out its primary function. So it's like the location where catalysis or function occurs. So the heme pocket for hemoglobin is the active site, okay, because that is where the ligand, meaning O2, that's where the O2 binds. Okay, so the heme pocket, that entire location, is the active site. Now, you have regulatory sites. Okay, so these are the allosteric sites that I mentioned. Okay, so basically, regulatory sites are the other sites on the proteins that bind to specific molecules which regulate the function of the protein. Okay, so allosteric locations where one molecule binds, alters the shape, and then cause the protein to increase function or decrease function. Now, commonly, binding of specific regulatory molecules at these sites will change the conformation. Okay, so usually it's some kind of a conformational change. Okay, and then that changes the activity. So the molecules that regulate protein function in this way are what we call effectors. And then they typically exert their effect through allosteric mechanism. Okay, so do you guys see why I care so much about allosteric effects? Okay, so those that increase protein activity are positive effectors. Those that decrease activity are negative effectors. So those should be pretty straightforward. You can also have homotropic effector, like oxygen in hemoglobin. That's homotropic. It means that this particular effector binds at the active site. Okay, and after you bind to the active site, it causes some kind of conformational change, and then it alters the protein's activity. Heterotropic effectors bind at the regulatory sites. So yeah, that should be pretty straightforward. All right, so first, we have the Bohr effect. All right, so we know that in your muscles are going to be primarily the acidic aspects. What we have is a decrease in blood pH results in a stabilization of the deoxy state. Okay? Therefore, it favors greater O2 release. And this response of hemoglobin to pH is a process we call Bohr effect. Okay, so this is very useful. Okay? This is actually very useful because as you're doing exercising, and then your muscles don't have enough O2, so it's going to start metabolizing its own sugar, and it's going to make lactic acid, right? You guys know that. 肌肉会做乳酸嘛, huh? Okay, and then making lactic acids lower the pH, right? And then as a result, here it says, lowering the pH, your hemoglobin will have a larger load, okay? A larger unload. Okay? So it's going to stabilize the deoxy state. Okay? So it's going to favor the T state. So Bohr effect, okay, so protons favor T state. So a lowering pH prefers T state, so it's going to make your protein go to T state and unload the oxygen. So as oxygen is utilized in tissues, carbon dioxide is produced and must be transported back to the lungs or gills. And the accumulation of CO2 also lowers the pH. Why is that? Okay, because there's an enzyme, okay, called carbonic anhydrase, and actually you don't need an enzyme for this, which takes CO2, react it with water, and you form bicarbonate and protons. Okay, so this is what sort of decreases the pH. By the way, erythrocytes are red blood cells. At the same time, the high demand of oxygen, especially in muscles involved in vigorous activity, can result in oxygen deficit or hypoxia, which lowers the pH by the production of lactate. That's what I mentioned. So the falling pH in tissues and venous blood signals for demand for more oxygen delivery. Okay? And then hemoglobin is able to do this through its allosteric transition between the high affinity to the low affinity, so between R state and T state. So why is it that lower pH stabilizes the T form? Okay, so we know that the T state is favored when you have a lot of protons, right? So at low pH, your histidine 146 is protonated. Where is histidine 146? This guy. And which one is protonated? This guy. Okay? Do you know what atom is this? 你们知道这个 atom 是什么？什么东西可以被 protonate? I mean, come on， 在 biology 里面有什么东西可以被 protonate？ 你也就做3个选项 ：carbon、oxygen 跟 nitrogen。Carbon. Anybody says carbon is protonated? No, not carbon. Carbon doesn't like to be protonated. Oxygen, nitrogen. Okay, why? This guy is histidine. Okay, so histidine, remember, is that thing, right? So it has a nitrogen, right? So your histidine is going to be protonated, and because it's protonated, it can form a good salt bridge with aspartate 94. Okay, so with this guy. So here you have O minus, and then here you have a proton. They can have very good charge polar interaction. Charge polar interaction, very nice. And then it stabilizes the T form of hemoglobin. Okay? And then this feature allows for hemoglobin to release more O2 and then also capture proton. Okay? Because your histidine has to be protonated. And by protonated, it means it captures proton. Okay? All right. Okay, so proton affinity is also higher in the T form. Okay? So at the T state, your histidine 146 is close to the aspartate 94, okay, and forming a salt bridge that increases the pKa of your histidine 146, okay, by stabilizing the proton against deprotonation. So what that means is, remember we said typical histidine imidazole is like around 6 or something, okay, but if you go to the T state, the pKa will actually become 7.9. And then 7.9 is closer to the physiological pH, okay, or the physiological condition. Okay, in addition, here we say your cell is producing protons, right? And then you have two choices. One is 6.4, one is 7.9. Which one is more likely to capture the proton? You can have a increase in pKa, essentially.